This is a production of Cornell University.
Well, thank you for the introduction, Margaret and Gaurav and other people who invited me here. Can you hear me in the back? Jerry Davis was reminding me earlier, the last time I was here and in this room was 2005, and I lost my voice and I whispered my way through the entire presentation. And apparently, I thought, well, hopefully that won't happen today. So since 2005, I've been at the University of Missouri, and it took me probably 10 years to figure out how to talk to people about my research. I used to tell people I worked on heterosis and hybrid vigor and chimeras and all these kind of things. But I finally learned if I really wanted to talk to people out in the public, it was the best thing I could do is say I worked on the dogs of the plant world. Because I could tell them that, you know a Chihuahua and a Great Dane are a dog, but did you know that broccoli, cauliflower, kale, Brussels sprouts are one species like a dog? And then we can spend the rest of the time talking about their dogs. And I'm just a nice professor talking about dogs. So that's first knowledge bomb of the day for people, is talk about your dogs. Now, if I get on an airplane and I don't want to talk to people, I just tell people I'm an evolutionary biologist. And that pretty much stops the conversation flat. Now, or I put on my headphones and start reviewing manuscripts. So, but if I am thinking about evolution, I really start with Ohno, 1970, and before that, because Ohno postulated that gene duplication played a major role in evolution. And so in the era of genomics, I think the big update to this is what I'm just going to tell you is the thesis of my talk today. And that's that all duplications aren't alike. So if you spend a lot of time like I do working with computer scientists, you really have to get them to recognize the difference between tandem duplicates and polyploid duplicates, because they behave differently and they operate differently in terms of systems. And so you have to do this lesson building. The second thing, which is going to be the theme of my talk, is these whole genome duplications occur throughout most of life, taxonomically, and also over time. And so I'm going to talk today about both recent and ancient polyploidy. And now that we no longer rely on reference genomes, but use pangenomes to understand variation, this structural variation is super important. So if you're doing GWAS, you can't just do it on SNPs. You have to include structural variants if you really want to understand the genotype-phenotype map. And that's just not for plants and crops, but also for human diseases and things like cancer. So this crowd doesn't need to know about what is polyploidy, because you guys all know what polyploidy is. But I do want to make the distinction between auto and allopolyploidy. So if you're a historical cytogeneticist like a Stan Peliquin, you defined auto and allo based on chromosome pairing. Whereas I'm going to talk about it today in terms of ancestry. And what I'm going to talk about specifically today is allopolyploidy, where you have the merger of two distinct species that come together. So another way to state this is I really like chimeric things, hybrids, and I'll even say grafting for Margaret's sake. So bringing two things together that normally don't belong, and how do you get novelty from the combinations of new things? So I would say the major advance since I started grad school in 1994 and finished in 2000 was knowing that from cytogenetic evidence, things like corn were probably paleopolyploids from compared to sorghum based on even basic observations. But the biggest surprise, hands down, from the Arabidopsis genome paper wasn't all the things that were promised when they started the Arabidopsis genome project. It was this figure and the other analyses that showed that this tiny little genome with 5 chromosomes had not 1, not 2, but 3 polyploid events, which were named alpha, beta, and gamma from the most recent to the ancient. And at that time, people speculated that alpha was about the base of the mustard family, beta was at the base of the eudicots, and gamma was at the base of all angiosperms. And all 3 of those assertions were wrong, which became the basis of the first part of my talk. So as we sequenced grape and ambrelle and other things, well, at least when I started as a grad student, we would say things like, oh, 70% of flowering plants are polyploid. Well, that was based on the idea of thinking about what a neopolyploid is and stomates and things like that. But if you now look deep in time about how you define polyploidy, we now know that all flowering plants, extant flowering plants, including ambrella, have an ancestor that was polyploid. So we really shouldn't ask ourselves, are you polyploid? We should really say, when was the last time you were polyploid? And so us who are vertebrates, well, the last time we were polyploid was about 350 million years ago, right? So we are polyploid, right? And people like Ethan McSlight had made a career studying how those polyploid duplicates left from the origin of polyploidy from vertebrates are now part of our disease network. And of course, there's a whole groups of invertebrates that are polyploid, as well as the most speciose groups of vertebrates like fish and frogs. So if you're in one of the 4 groups of this school that works with crop plants, and you care about domestication, you should also know by now that crop plants are, compared to the wild plants, more likely to be polyploid. So one question is, how do genomes and all the other omics and networks evolve in brassicas or any crop based on this knowledge? So the very important plants I'm going to talk about today are brassicas. So the dogs of the plant world I call oleracea, cabbage, broccoli, cauliflower, so forth. Brassica rapa, maybe you don't know, is also a dog or cat, because it has all these amazing Chinese vegetables, things like turnip. And Brassica napus is also pretty diverse, has underground things like rutabagas or Swedes, leafy things like Siberian kales, and then a whole suite of oilseeds. But here's the real big picture of what my lab's been doing. So my lab, along with, I would say, Doug and Pam Soltis' lab and maybe Jeff Doyle's lab, a few others, have been looking at polyploidy not from recent times, but also at the base of the families and all the way back to the origin of flowering plants. And so what I'm showing here is brassica. If you imagine, we'll start on the left. So imagine that in the mustard family, you had an 8 chromosome ancestor. Over evolutionary time, Arabidopsis became diplodized to 5 chromosomes. And then somewhere time around 15 million years ago, there was a hexaploidy, so two rounds of polyploidy, a hexaploid event. So you went from 8 to 24 chromosomes in the brassicas. And then those diploidized down to 9 chromosomes in oleracea, 10 chromosomes in rapa, 2 to 4 million years ago. And then those merged together to make Brassica napus and allopolyploid. And so this is neopolyploidy, Brassica napus, mesopolyploidy, this kind of where you can see chromosome segments and blocks. They're acting like diploids, but you can still see these blocks. And then paleopolyploidy, the alpha, beta, gamma event, and Arabidopsis, where you just have duplicate genes left, but no synteny really among them. So I'm going to give my talk today talking about these 3 polyploid eras, if you will, going from oldest to most recent. Oh, by the way, if you add up all the polyploid events together from the origin of flowering plants, Arabidopsis is 12x, the diploid brassicas are 36x, and Brassica napus is 72x. So when we published that genome in Science in 2014, it was the most polyploid genome at the time. Okay, those are the 4 parts of my talk. Okay, so yesterday, day before, is Fei Wei here? So Fei Wei and I and 200 other people over the last decade have thrown together 1,000 some odd transcriptomes under the leadership of, I would say, mostly Jim Liebensmack and some other people. And we spent a lot of time on this project, kind of like the Arabidopsis Genome Project, to make a big tree, a big nuclear gene tree of flowering plants to contrast to the mostly chloroplast organella tree. And we also wanted to look at gene family expansion. But what was the big result? It's like the Arabidopsis genome result with its 3 polyploid events. The big result of this paper has nothing to do with the tree. It really has to do with this. 244 ancient WGDs, whole genome duplications across green plants, 65 that match published genomes. Every land plant lineage has an ancient polyploid event, except for some weird ones, liverworts. And by contrast, almost none of the things in the water, algal lineages, have evidence of polyploidy. That, in my mind, is the big result. So now you don't have to read the paper. Just came out 2 days ago. You can go with that. Okay, now I want to read you the last 3 sentences. So here it is. Gene and genome duplications have been considered a source of evolutionarity. However, the direct connections between key innovations and specific gene duplications are rarely known, due in part to lag times between duplications and such innovations. So earlier today, I met with Gaurav, and we're gonna, I say we, he is gonna explain this, because the second the genes and everything else duplicate, yeah, enzymes don't change right away, right? It takes some time to do stuff. So if you look on a tree where polyploid events occur, there's always a lag between when they actually get novelty and when they actually speciate. So anyway, we'll be talking about this in the future. So and then what do they say next in this paper? We need to do more experiments on this. So in my mind, the way that you do these experiments, not just in one system, but across the clay that has polyploidy, be it grasses, andropogony, or brassicaceae. Okay, so starting with the oldest polyploid events. This part of this project was motivated by what's the origin of mustards. So you know when you eat wasabi or mustard or radish, any of these spicy things, they don't have those flavors and tastes because they're good for us. It's because they're good for the plant, right? It's keeping away critters. Well, they keep away a lot of critters, but other critters, like these butterflies, have learned how to eat them. And so Then what happens? Caterpillar shows up, nom, 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 eats the plant, and the plant goes, help. And then here comes some little wasp or something else. So you have these tritropic interactions, and there's microbes involved and everything. So you have this really cool evolutionary relationship, which this relationship alone is how Ehrlich and Raven coined the term coevolution, was thinking about this model. So because I told you earlier about alpha, beta, gamma, people didn't know where this beta, gamma event were. They thought that the one was at the origin of eudicots, another one beta event. I was at a meeting in China, and Mike Fleming was there, and he said something he probably should have known, which was, oh, Andy Patterson and I are just looking at the papaya genome, and it doesn't have beta. And so Pat Edger, a grad student in my time, who's now a professor at Michigan State, said, we're going to find beta. So that's how this project started, was we're going to find beta, and then we're going to see an alpha, these two ancient polyploid events in Arabidopsis, and we're going to see how they affect the glucosinolate network. And then Pat defended his PhD in 2012, and we're going to publish the paper just on the plant side. And at that time, Chris Wheat was driving across Missouri to go to Kansas to talk about the Lepidoptera butterfly part of the story. And we lined up these trees, and it was too good to be true, so we got to publish this paper together. So then this is the only time in my life where I said, oh, 50th anniversary of Peter Raven and Paul Eric's paper, blah, blah, blah. It's got to be a Nature paper. No, no Nature paper. Science paper. No, no science paper. So we finally published in PNAS, and it's been our biggest by far thing. So NPR, Washington Post, Radio New Zealand, Daily Mail. But I was still not too happy about the Nature Science thing until it was the opening joke of a Big Bang Theory episode. And then I said, screw you, Science and Nature. I got a jumping joke on a Big Bang Theory episode. I don't need your stupid journals anyway, and I'm not even going to try to submit there anymore. So without further ado, I will tell a brief version of the story because I told it many times before. So what happens, right? Plants evolve a novel biochemical defense. The plants radiate. We showed that. Insects evolve a counter-adaptation. The cool part of this story, the butterflies don't have polyploidy, but they have a tandem gene duplicate that gives them basically the countermeasure to the mustard oil bomb. So then the butterflies diversify, and then so forth and so on. And now the butterflies, of course, are cued into what is necessarily the defense of the other plant. Okay, so the big result of this is shown in this figure. So this is first thing we did was we made the first nuclear gene-based phylogeny, the Brassicales, before it was unresolved. So we could actually do what hadn't been done before, which is map all the glucosinolates on a tree to infer the order that they evolved. The second thing was we showed where the polyploid events were based on transcriptomes and shallow genomes. So there's the blue diamond for beta and that blue diamond for alpha. So we showed where those polyploid events were. The third thing was we compared where the polyploid events were relative to species radiations. Those are where the stars are. So the Brassicaceae and those top ones in green, the three big speciose families, are totally different than all those other families. If you're taking plant systematics, you probably don't learn Pentadiplandraceae in plant systematics. Well, maybe you do here, but most places, no, there's only one species. So then the other thing was the innovation of the glucosinolate classes that correspond to these polyploid events. So there's where the radiations occur. And so we identified that there's these novel pathways, and similar work has been done in the Solanaceae by Rob Last and other people. So since then, this work has continued. So we've been updating the kind of phylogenomic framework for the Brassicales, thinking particularly about glucosinolates. And we just posted on bioRxiv a paper that looks further into other polyploid events, such as within the Cleomaceae and some of these other families. And next slide. We now have this really large three CSPs with JGI to look at not just the genomes, but also the transcriptome and metabolic profiling across members of this group. And so we have massive convergent evolution of various metabolic pathways, polyploid events. We have 19, 20 new genomes to add to ones that are already out there. There's this huge team, including Jacob Washburn in this room and others who've helped with this. And these recent projects are, we're going to go deep into the Brassica oleracea, Cranberry hispanica, Philosophy arvense, which you may know as pennycress, camelina, and Arabidopsis to do like platinum genomes for about 10 to 20 sessions and to do a whole bunch of tissue-specific profiling of transcriptomes and metabolomes to see how those change those networks. And Jen Weiss-Kaber and Pat Edger and Dan Klebenstein and I are leading that effort. Okay, so that's ancient polyploidy in a nutshell. So old duplicates really matter for big, novel, key innovations that can result in classic and clade-defining features. So what about these polyploid events, which you can't see under a microscope, but if you paint a genome, you would see like, oh, for every back-sized piece in Arabidopsis, I see 3 of those and Brassica rapa or Brassica oleracea. Okay, so here's a simple slide showing the diversity of Brassica rapa. You have things like turnip, which you're probably familiar with. If you're in any Asian market or farmer's market that has Chinese vegetables, you have Chinese cabbage and bok choy. You have sarcens or oil seeds, which come in yellow and brown colors. You have some other field mustards, and you have this interesting thing, rapini. So I'll get to that in a second. So when we started this study, and this was a collaboration with Mike Gore and Joanne Labatt that started with for some GBS sequencing of the USDA collection. What do we know about Brassica rapa? Well, there were two studies that were out. One was in 2011 about how there are basically some morphological groups. There was a study in 2014 which said actually there's geographic groups because things like turnips are found in Asia and in Europe. And so turnip-like things are more closely related to things that look different than they do together. And then while we were in the middle of our study, there was a large paper by Cheng et al. On both rapa and oleracea, which gave out this tree, but as you can see, the groups are what we found, but it's pretty comb-like in how things are related. And so what we wanted to ask was, could we tell were things domesticated multiple times, were they moved by people, and so forth? And then the real question that was burning for me and also for some other students was, what about these leafy things? Are they wild? Are they domesticated, or are they feral? And so in this first study, we had a lot of accessions, 333, and we found these major groups. So we found bok choy as a group on the top, Chinese cabbage, and they weren't formed by hybrids, which some people had hypothesized. We found Asian turnips and various other Asian vegetables in purple. The sarcens formed one cluster. So yellow and brown sarsen are really just one thing. They're just different by a gene or two. And then we found these turnips at the base. And also some groups that are named taxonomically like field mustards or polyphyletic. Now the problem with this study, so we're the first ones to show this, but the problem in this study is we didn't sample rapini and a bunch of other types, and also there weren't really any wild samples that were put in this. So then in a follow-up study, this was a collaboration with Mike Barker and Gavin Conant and I on a planned genome grant. We looked at the diversity of Brassica rapa using transcriptomics. So our lab is one of the labs, along with Claude DePamphilis, Jim Lehman's-Mack, and others who really got off to an early start using transcriptomes, both for phylogenetics and population genetics. So this was looking at 126 successions. We again found these same groups as I talked about before, but we did answer one question, which was the rapinis. Pointer on it. The rapinis here were clearly fully nested next to these sarcens, so they're not at the base of the tree. So that was like the big result from this paper in terms of how the topology of these groups looked. But we didn't sample Asian turnips and a bunch of other things. The other thing that we did in this paper was this was the first time that a demographic model like DADDY had been used. So some people who worked on those in humans helped us work on these in these plants. And the two postdocs working on this project, Jinshuai Qi in Mike Barker's lab and Home An in my lab, both were into reading about where cabbages and things are in the Chinese literature. And so they're able to track that, oh, where we saw the origin of turnip was where the cuneiform tablets had them on in Babylon and Iraq. And where they were in classic poetry in China 100 years ago, or 1300 years ago, or 960 years ago, pretty much matched in a very rough sense when these demographic models put the spread of Brassica rapa mostly from east to west. So it turns out, of course, that follows the Silk Road pretty nicely, and there's lots of admixture back and forth. And in contrast, there was a bottleneck at the origin of the Saracens, and that happened about the same time as when Alexander the Great went into that part of India. So that sounds like it's a very nice and neat story, except for this guy. So this is Alex McAlvey. He was a grad student of Eve Imschwiller's. I think, Jeff, was that one of your students? Eve? So this is Jeff Doyle's academic grandson. He's now a postdoc here with Sunny Powers, I think, and he's been collecting a ton of these around the world. He says that's a really simple story. There's actually 13 different subspecies of Brassica rapa that you folks are missing. There's all these feral things all over the world. And actually, his hypothesis is that Brassica rapa originated probably in the Caucasus and spread east and west. And so anyway, there's a lot more to do with this. It's not a done story at all. All right, so back to the network idea. So with this kind of population genetic framework of how Brassica rapa has evolved, what about these duplicate genes? And so Mike Barker and the consortium of us have a manuscript that we're putting together where basically we're looking at targets of selection with a particular comparison between duplicates that come by tandem versus polyploid. And so looking at the paleologs for these, we asked this question: do paleologs have a higher genetic diversity than other genes of Brassica rapa? And the answer to this is yes. There's a fourfold higher diversity of these paleologs. So now what I really like to do is take these lists of paleologs and look at them not just in Brassica rapa, but also in Brassica oleracea, like kales, or in arugula, or other plants that are related to this event. So I like to think about I like to ask people not just what their 5-year plan is, but if you could summarize your research into one question, what would it be? So this, I don't have a one-word answer, but I have this picture. So in my mind, I'm really interested in how networks evolve over time and what happens when you're a chimera. So if you imagine any network on the left side, after whole genome duplication, you have this plethora of possibilities. And over time, you have mostly gene loss, but you can have novelty by having new topologies of relationships, which is indicated by that arrow in red. And my thought partners in this over the years have been Jim Birchler, Gavin Conant, Mike Freeling, and others, to think about, well, if you wanted to understand human medicine, wouldn't it be cool if you had a polyploid human, right? So if you want to understand a plant, wouldn't it be cool if you had a polyploid plant? Oh my gosh, we have polyploid plants. So when you double the system, can you really look at the constraints of network evolution and what the consequences are for phenotype, crop, synthetic biology? And so Gavin and Jim and I wrote a paper in 2014 on this. Jacob Washburn and Kevin Bird and Gavin and I wrote a paper in 2016 on convergence, and multiplied is about convergence. And we published a paper in 2018 on phylogenomics and network evolution. That is, we spent a lot of time on. And then Gaurav and I and a group of us published a paper recently on thinking about this in terms of metabolism. So if you want our philosophical thoughts, there they are. All right, so back to experiments. So this is an unpublished experiment on Brassica rapa, Arabidopsis, and Cleome. And we would have published it a long time ago, except the Brassica rapa genome that we used for it is still sitting unpublished in an unnamed group's basement. So I'll show you the results and then we'll just repeat it someday. So the experiment was this, was if you look at Arabidopsis and Brassica across tissues and across a bunch of different challenging treatments over a circadian time course, what do you see? So we're using light, dark, flooding, salt, cold, and methyl jasmonate. So imagine in a broad scheme, you have whatever Arabidopsis network that you want to put together. Brassica is going to have 3 subnetworks from, right, the whole genome triplications, hexaploidy. And over time, one of those subgenomes, LF, these fractionated, still resembles the ancestral Arabidopsis genome pretty much, whereas the other 2 subgenomes have become highly fractionated. So if you look at the genome of Brassica rapa, only 7% of it is still in triplicate, 32% of it is in duplicate, and 61% of it is single copy. So now let's look at some abiotic stresses. Only a fraction of the duplicates are expressed in any particular stress, whether it's hypoxia, salt, cold, or methyl jasmonate. But what I'll show you is that they're different duplicates in different situations. So in Arabidopsis, if your entire interactome is 10,811 nodes, in Arabidopsis, this abiotic stress node is about 961. And in Brassica, okay, and then in Arabidopsis, by the way, that stress network, 238 nodes, so 25% of those nodes go off whenever they get stressed. In Brassica, you have many more nodes from the subgenomes, 537, 289, 239, but only 2% of those respond in all conditions. So I was explaining this to some non-scientists, so I decided to make this cartoon. So I think of it like this. So Arabidopsis is like one radar to detect things, and if something goes wrong, it sends a fire truck every time. Brassica's got radar, sonar, you know, everything are, and it sends a fire truck, a helicopter, or a boat, depending on what kind of stress is happening, right? So Brassicas, or anything that has these kind of polyploid networks, have a lot more flexibility in the kind of things that they can do in either a sub or neo-functionalized context, let alone a dosage context. Okay, so to dive further into this, since 2013, we've made a pretty exhaustive catalog of concerned non-coding sequences among the Brassicaceae. So I'm going to show a couple cartoons where I have a gene that looks like this, where there's 2 green exons and a bunch of little CNSs of these little yellow boxes. So after polyploidy, this is what you often see. So on the top is an Arabidopsis gene, It's got 1, 2, 3, 4, 5, 6 CNSs, and it's got 2 exons. If you look in Brassica, almost none of them will look totally like that. But you have the one in the least fractionated genome will have all the exons and most of the CNSs. And then the 2 other subgenomes will either have a loss or have more CNSs lost. So what is this? So Ed, pay attention, this is your slide. So CNS loss is consistent with subfunctionalization following gene duplication. So you can use polyploidy and polyploid-aware AI to think about constraints in these kind of models. So whenever somebody comes and gives a talk, I got the latest and greatest CRISPR, high throughput, blah, 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 and I'm gonna do all these mutants. I'm like, well, you go ahead, I'm gonna spend 2 hours on my computer and figure out that 90% of those things you wanna do are crap, because evolution has already done the experiment. So for me, like when I look at like the Pan Am project or some of these other projects we have going on, we're gonna model most of the space out, right, to know where we need to go after for doing actual targeted gene editing. So use evolution. Okay, was that okay, Ed?
Yeah.
Are we good? All right, just checking. All right, so now it's just, so I have this data for like every gene that's in Arabidopsis that we know enough about. So here's just one cartoon example of fractionation mutagenesis so you can appreciate it. So on the left is an Arabidopsis gene, and on the right are the 3 versions of it in Brassica. So let's go look at some cells. And this is just crude tissue stuff, so of course you can see where we're going with this with single cell technology today. So if you put this gene under a couple of time courses, in Arabidopsis, in the morning it goes up, and then in the evening it goes down, right? Typical gene that's responsive to light. Oh, CNS6 is light responsive. So you'll notice over here what's going on in the Brassicas. So this has got its CNS thing. Here's CNS, so this one's gonna respond to light. So sure enough, this Brassica homologa, this responds just like that one. This one's dead, we don't have to worry about that. What about this one? Oh, it doesn't respond to light. So it doesn't do anything, right? No, of course it does stuff. So this Arabidopsis gene, it's like the fire truck, right? It shows up all the time, every tissue, does everything. This gene acts just like Brassica, all tissues, all the time, always on. This one, oh, shows up in the dark. Shows up when there's flooding, right? So this is the boat. This is the speedboat version, right? Okay, and then there's a whole like exhaustive gene, and the networks for this, so you get the idea. Okay, so take home tweetable point number 1. Who's tweeting? Thank you, Margaret. Good God, one person, F. So tweetable point number 1. You can now use these systems to think about, we used to use terms like neo and subfunctionalization, talk about a single gene, right? The outcome of a single duplicate gene. Now we can think about networks of these ways. Gaurav, yeah, are you on board? Mm-hmm. Okay, not sure. All right, so in my fantasy experiment, you have 1,001 perfect genomes of Arabidopsis, including tandem duplicates and polyploid duplicates. You have your 1,001 Brassicas, you have your 1,001 sorghums, you have your 1,001 mazes, and you can tie this together to really understand constraints. And Jeff, what did you call it today? Homeostasis. Why do we have, I mean, maybe the real question isn't why are things different, but how do they stay the same when they have all these ploidy levels on top of them and go through meiosis and make pollen and eggs? Okay, so I've been thinking about this a lot along with Jen Weisskaver and Gaurav and others about how could this affect both primary metabolism, things like CAM and C4, as well as specialized metabolism like glucosinolates or cardiac glycosides, or name your favorite metabolic network. Okay, good. Oleracea. So now we're on to the dog. So this part is completely unpublished. It's totally tweetable because we've talked about it before, but this is all new. Okay, so this work is, what I'm going to present to you today has mostly been by my graduate student, Mackenzie Mabry, on the left. This is part of, again, a USDA grant with Mike Gore and Joanne Labatt and some other people. But we also have Sarah Turner on the right, who's in now Jeff Ross-Barr's lab at UC Davis. So I'm going to show you transcriptome data, but Sarah's got genome data, and together they have all this other morphology and phenomics and ionomics and stuff that I'm not going to talk about. So today I'm just going to talk about wolves and dogs. Okay, so I love this new trend for de-domestication, because every time people write a review paper, they'll start with Zach Littman's tomato, they put in the review paper this picture. So I'm like, if I don't write the de-domestication grant for Brassica like the next month, I'm a fool, right? So, but there's a problem with this picture. 1, it doesn't show how those different things are related. And 2, that thing in the middle is fiction. There is no wild mustard plant that looks like an annual herbaceous thing, that's a Chinese white kale. That's a domesticated thing. So first of all, what the heck is that we're going to de-domesticate? Doesn't look anything like that. Okay, so who are the wolves and who are the dogs? So these are all the other things, by the way, besides the kind of 5 canonical cabbage, broccoli, cauliflower. You have, as I learned yesterday, favorite part of my trip so far, Margaret and Griff. Is Griff here?
He's in Geneva.
Oh, good God. So anyway, Griff, hi. We went out and looked at all these amazing, there's like more diversity in this field of kales than I've seen in my whole lifetime. And so there's all these different kales and collards and walking sticks, like phenomenal diversity. Like, who cares about dogs? These are way cooler than dogs. Okay, so what's the progenitor? All right, so here's some possible progenitors. So unlike Brassica rapa, which occurs on the continent, Brassica relatives, wild relatives in the sea genome, which also have 9 chromosomes, are mostly in the coast of the Mediterranean. So they got these like kind of really hard leaves. They go like, looks like they're like, can take salt spray. They're woody at their base. They're perennial. And these are all the possible contenders for who could be the extant closest relative to Brassica oleracea. Okay, so there's been 3 competing hypotheses out there. So if you're British, you like the hypothesis that the origin is in Britain. Is that a surprise? No, and so why would you think that? Well, if you go to the coasts of UK, you find things that look like wild cabbage. They're also on the coast of France and Spain, but that British don't care about that. So you think, okay, well, British, wild cabbage. What's another? So if you like the Vavilov arguments, which is also false, but like if you like the argument where there's the most number of species, that should be the origin. So the most number of wild sea relatives are in southern Italy, particularly Sicily and Sardinia and Corsica. And then recently, people have done phylogenetic work not on DNA, but on the words. So if you do a phylogenetic analysis of the words of the species, Brassica cretica, which is on Crete, and Brassica hilarionis, which is in Turkey and neighboring area, would be the progenitor, sorry, not progenitor, wrong term, the extant sister species to domesticated Brassica oleracea. So those are our current 3 hypotheses. So let's see what we find. So we started this project, and again, this really nice paper, Cheng et al. Came out that shows this relationship where you have cabbages, kales, Brussels sprouts, kohlrabis as one group, cauliflower, broccoli, is another group, and then Chinese kale, sister to the rest of those, but not really a good analysis of what was sister to it as far as the wild things go. Okay, so I have 2 questions I want to pose here. First, how are the wild relatives related to each other and to Brassica oleracea, and where they occur? So who's the wolf? And then how are all these dogs related to each other? Okay, so this is, I'm only going to talk about, I'm going to show you kind of PopGen phylogenetic trees today, but here's the data that we collected, and we've done this for rapa, oleracea, napus, and some other things like arugula. We grow plants in a growth chamber, we put them in 4 replicates, we have all of these leaves, we have kind of a semi-high throughput way called undergrads who harvest these things and do little vials. And so, you know, leaf 4 is for RNA, leaf 2, 3, and 4 is leaf scan, leaf 2 is for metabolomics, leaf 3 is for ionomics, and we have all this data. One undergrad, Brandon Geinstraub, really got into Raspberry Pi, and so he made these like Raspberry Pi system for looking at it. So we published a little paper on this. Like, this is like a pretty thing, so you wonder how you get 150 papers. You have a good grad student like McKinsey mentoring undergrads to publish little papers like this along the way about a method like Raspberry Pi, right? Totally cool undergrad project. Okay, so now some results. So here's the first PCA. I don't think Michael has seen this yet, but anyway, the first PCA here is on the right are wild, on the left are all the domesticates. And so who are these guys here that are the wild ones that are the closest to domesticates? Okay, they are Brassica cretica and Brassica incana. Okay, so let's look at a phylogeny. So this is the pretty typical these days in phylogenomics. If you look at this tree, it has 100% bootstraps. And you can run a different program, and it'll also have 100% bootstraps, but it'll be a different tree, right? So bootstrap values are kind of meaningless in this era when you have 100% bootstrap for conflicting relationships. So that there's a whole other conversation to have about that. Okay, so what we find are a monophyletic group of domesticates, Brassica cretica, sister to those, Brassica incana, sister to those, and then some alternative topologies, they flip, or they're both sister to the domesticates. And here's the other thing. So sorry, my friends in the UK, but your wild cabbage on the coast is not wild. It's not a wolf. It's a feral dog, right? It's a feral dog. Okay, so that's what you got there. And then within the domesticated plants, the domesticated brassicas, you got collard greens and cabbages, and then kohlrabi, cauliflower, and broccolis. And we got lots more to do in this. So I told you that. I told you that. Okay, so where are we going in the future? So we have all this other data to use, not just for GWAS and TWAS, but we have all these glucosinolates and things to work on. We have morphometrics and leaf scans to play with, and we may also work with Dan Shitwood or some other folks on this. And then finally, the reason why I came here was to go out yesterday and look at the huge diversity set. So we have this amazing GWAS platform. It would be so easy to add some of these other things that some of the students here have been working on for Brassica, and that would be fun to do together. And also for machine learning, from everything from phenotypes to enzymes, like there's a lot of things that would be fun to play with people here elsewhere. And my last little piece on this is on fertilization. So I like to think of polyploidy as a window in the genome evolution, but I've been thinking about these little feral dogs as ways to think about domestication, because it's like domestication reverse. So if we have these woody cabbages off the coast of UK, France, and Spain, well, guess what? They're also off the coast of California. So when I was an undergrad at Berkeley in 1986, Herbert Baker would talk about these wild cabbages that are now off the coast of California that have they become woody again, right? And so like this is like a huge project just sitting here to look at the parallel de-domestication back to woodiness and perenniality across these different habitats. Well, and by the way, if you go to the islands of, Canary Islands or Channel Islands off the UK and Europe, you get walking sticks. These are 8-foot tall, woody, so if there was still money for Arabidopsis biofuel money, this would be the thing you'd want to work on. But that money is not here anymore, so that's too bad, damn. Okay, so how can we use this de-domestication fertilization process to understand crops in a rapidly changing environment? Okay, so last part, how am I doing on time? How much time do I got?
You're good, you have like 15.
Oh, good, okay. So last part. So this is, normally I do this thing switched. I do the neopolyploids and end with the big. So this time I did a backwards experiment. So now I'm going to talk about what happens right after you make a polyploid. So Brassica napus is classic heterosis, right? So on the left is a Brassica oleracea rapid cycling plant. It's about this tall, flowers in 30 days or so. On the right is Brassica rapa, fast plant, right? Wisconsin fast plant, flowers super fast. In the middle is Brassica napus. I think Birchville has his mini maize. This is our version of mini maize. So, but Brassica napus is things like this tall. Commercial canola is like this, right? This thing is like this. But we can do, when we first made them, we can do 2 of these a year. So the origin of this experiment, I don't know if you remember, but Lauren Riesberg became famous for making 3 hybrid sunflowers, 3, maybe 5, and comparing them to a natural sunflower and showing that evolution repeated itself when you replayed the evolutionary tape for chromosome rearrangements in sunflower. So Tom Osborne, where this project started in Wisconsin, in agronomy, saw that experiment. He's like, 3, 5, let's do 100. So we made 100 resynthesized Brassica napus lines, but this is not in the era of next-gen sequencing. This is the era of RFLPs, and I could put 50 on a southern blot. So that's why there's 50 and not 100 in this study. So over time, what happened is, this is just a portrait of one of the lines. I self them, and by generation 1, 2, 3, 4, 5. So this one became dwarf, right? Other ones became early flowering. Other ones became late flowering. So even though they start off with the same exact homozygous parents, double haploid, we got this huge change in phenotype. So how could that happen? They had homozygous parents. So we discovered some stuff, and I'll show you. So that big experiment was actually first thought of from one cross. So in 1995, Kaming Song and Tom Osborne published a paper where they made some crosses. Now they weren't self-plants, and they weren't, but they showed dynamic changes, and a lot of people didn't believe them. A lot of people didn't believe that result. So this was a more careful experiment where they had inbred lines of rapa and oleracea, and this is so Jeff, was Eric Schranz your undergrad?
Yes.
So Eric Schranz, who was an undergrad here at Cornell in Jeff's lab, came to Wisconsin. I think he actually wanted to work with Ken Seitzman, but he ended up in Tom Osborne's lab doing Brassica stuff. So this is what Eric did. So Eric, and the other nice thing is right down the hallway is Rick Amasino's lab, which is like a famous lab for working on flowering time network in Arabidopsis. So Eric had the smart idea to glue what Amasino's lab was doing in Arabidopsis with FLC and Constans with what was going on in Brassica, because there'd be 3 copies, right, of these flowering time genes. So Eric started to do this experiment. We made these crosses and selected for early and late flowering. And after 6 generations of selfing, they're basically 2 species. If you throw the seeds out, the early flowering ones would never see the late flowering ones. So how'd that happen so fast? Well, and by the way, this was about the time when Hopi Huckster and other people were making a lot of hay about speciation genes. So everybody wanted to find the speciation gene. So we didn't find a speciation gene. So then we did this big experiment. We made 50 lines that were started, so 50 independent lines. There's not one line, there's 50 independent lines. And we self them, and we published the big paper, and this was in 2007, and we followed up in 2011, and I'll show you some new stuff next. But when you first made them, they were all identical, but after even 5 generations of selfing, they were really diverged, both genetically, epigenetically, and phenotypically. So here's, I think this is the only slide I also showed in 2005, but I always show it again and again. So this is my most important slide. And you have to learn how to spell homologous after this, okay? So these are rearrangements caused by homologous recombination. So, you know, in a diploid, so in green is Brassica rapa diploid, yellow is Brassica oleracea. They have homologous, 1O, chromosome exchange. And the allopolyploid, they have homologous, you have to have 2 vowels, not 1 vowel, 2 vowels, homologous chromosome exchanges. So if you ever get that reviewer comment, how to spell homologous exchange, that was me, or Jim Birchler. So as a result of homologous chromosome exchanges, you can have, so the exchanges are reciprocal, right? The exchanges are reciprocal, but as a result of a Punnett square, you can have non-reciprocal inheritance, okay? So you can get results that look like this, and you can also get reciprocal exchanges. Okay, so these rearrangements can have huge effect on dosage, and we call them speciation rearrangements at the time. So imagine that these bubbles are a phenotype. So let's say FLC and constans are here. Brassica rapa in the big circles flowers in, say, 10 days. Brassica oleracea in the little circles flowers in, say, 30 days. And the mid-parent flowering time for napus is 20 days. You have a chromosome rearrangement on the one on the top and the one on the bottom. You have the 2 separate flowering times, right? And you can do that for any pathway, right? Whether it's flowering time or glucosinolates or whatever. So does this happen a lot? So we published this result. And again, just like the 95 paper result. So we don't believe it. Oh, now we believe it, but it's only true in your resynthesized lines. It never happens in nature. So then we sequenced the genome, and in the genome we found 17 homologous exchanges. And then people said, okay, well, that's fine. It only occurs in resynthesized brassica, your crazy crop brassica napus, but it doesn't happen in any other crop. Well, then after we published this paper, it was found in wheat, and then strawberry, and then tragopogon, and a bunch of grasses. So if you don't know about homologous exchanges and you're doing pan-genome work across any group of plants, you should really think about this as a source of variation if you're now looking at copy number variants or presence-absence variants. Should you care if you're a plant breeder? This sounds pretty obscure to me. I don't, who cares about these homologous? Okay, so here is a picture of the homologous exchanges across winter canola, Darmore, Bristol, spring canola, Aviso, some Japanese and Korean types, a synthetic type, the kale one that I'll show you next, kale type napus, and a Swede, rutabaga. All those colored boxes are homologous exchanges. And I just highlight one in green, that's glucosinolates. So glucosinolate evolution, totally partly at least responsible by these homologous exchanges. And then we also see these for all kinds of other things and now in other crops. So these 50 lines haven't died, so we're still replaying the evolutionary tape. So this paper was posted on bioRxiv, I think 2 days ago. It's actually called Replaying the Evolutionary Tape to Investigate Subgenome Dominance in Allopolyploid Brassica napus. So Kevin Burge, Had Niederhuth, Malia Gehan, a bunch of other people led by Pat Edger and Rob Van Buren. They looked deeply at 6 of these 50 lines, and they looked at methylation bias, homologous expression, and what are they finding? Guess what? Repeated patterns. So it really matters if you're thinking about constraints, what happens, who are the parents, who's the mom, and who's the dad, and you'll see the same things happening over and over again. So again, you don't have to CRISPR out everything, you can learn something from evolution. So, okay, Margaret, are you my Twitter person? Okay, here's Twitter point number 2. So you can find, by making synthetic allopolyploids, you can see things happen faster, like homologous exchanges, so you will believe them, because you see them faster and over again. You don't think it's an experimental error. You actually think, oh my God, homologous exchanges actually happen. They're not gene conversion events, they're homologous exchanges. I mean, and then you don't see them as being weird, you see them as something that are real, and then suddenly you can see them not just in brassica, but in wheat, and peanut, and a whole bunch of other things now. Okay, so what about the history of brassica? So Brassica napus, of course, is known as part of the triangle view, originated from the combination of Brassica rapa, the AA genome, and Brassica oleracea, the CC genome. So long-standing questions, which of these vegetables are the parents? And then once you get Brassica napus, you have further diversification into oleifera, which is a rapeseed type, subspecies rapifera, which is rutabaga or Swede, and pabularia, which is Siberian kale. So if you think about multiple origins, Are these different? Are these different morphotypes different multiple origins, or do they diversify from the same thing? And then finally, you have big differences in both winter and spring types. And so when we started this project, we had reference genomes for Brassica rapa, Brassica oleracea, and Brassica napus, and we did a massive transcriptome and now resequencing study, but I'm just going to show you the transcriptome stuff today. And so the big question for this, and this is a study led by Homan, he came to my lab from Wuhan as a visiting PhD student and has now stayed on as a postdoc, and this paper was just published recently in Nature Communication. His big question for his PhD project was, is it true that there's multiple origins of Brassica napus, because that's what's in the literature? And if it's true, how did multiple origins happen? Do the rutabaga Swede types evolve from things like turnip and kohlrabi? Do the leafy Siberian kales evolve from things like the leafy brassicas? And do the oil seeds evolve from things like the sarsens? Or were there multiple origins to one thing, like an oil seed type, and then they diversified? Or was there a single origin? So this is the three options for what he was pursuing. Okay, and so we got a global collection of Brassica napus, and unlike a lot of other studies, we included rutabaga and Siberian kale. We did like some really nice, generated a lot of really nice RNA-seq data and GSS data. And here's the, for those of you who are interested in methodology, here's the methodological take home at this point. It really mattered whether you used a reference genome, a single reference genome, or whether you used a pan transcriptome or a pan genome. The groupings weren't different in structure, but the phylogenetic trees were really different. So if you're thinking about that would be what I think is the big take home of this paper. And so there's home, and this is the paper that I'm going to be talking about for the next few slides that just came out. Okay, so the first big result was we discovered a new group of Brassica napus. So we have the typical rapeseed types in Europe and North America and in Asia. And then you have spring types. And then you have Siberian kales, and you have rutabagas. And then there's this interesting little group I'll come back to later, intro S, right here. Okay. Now, if you make a tree, so this is where it gets wonky. So we always get these four monophyletic groups. So rutabaga, Siberian kale, winter rapeseed in East Asia, and spring rapeseed are always one group every time you look, no matter how you look. These other winter rapeseeds that occur in Europe and America and Asia are a paraphyletic mess at the base of this. And then the relationships among that paraphyletic mess and all those four monophyletic groups really vary depending on what methods you use to make this do this kind of tree building exercises. And there's also network versions of this, but I'm just showing the cartoon version of it today. Okay, so I've been thinking about, we started with this Brassica napus, who's your mother? So there's a baby bird that runs around asking all the other animals about who's their mother. We still don't know who the mother is because I don't think we have the right diploid extant relatives. But then my Chinese postdoc says, we know that story isn't really an original story. In China, we have the story, except it's a tadpole. So he showed me these stamps where the tadpole runs around and asks the fish and the crab, and who's your mother? So I didn't realize this is one of those cross-cultural stories. Are you my mother? So we haven't figured this out for napus either. All right, so just to finish up what this paper had in it. So we found these 6 Brassica napus groups. There's a ton of admixture across them, particularly the oilseed varieties. We can't confirm what the general tale is, that multiple origins happen. It seems that's actually more like backcrossing. The nuclear and organelle phylogenies actually showed that intro S group that we discovered is so in China, they used to grow Brassica rapa for their oil seeds. And after World War II, they grew Brassica napus, but they backcrossed it to Brassica rapa for disease resistance and other things. So that's what that group is just the introgressed Chinese group that's got these other ones in it. And then I didn't talk about it today, but basically we looked really hard for all the selective sweeps we found, focusing on rutabagas and Siberian kales because everybody else is looking at oil seeds. We have a lot of phytohormones to chase down and other kind of metabolic things to go after. And just like with Brassica rapa napus, there's this obvious subgenome, both physical asymmetry and expression asymmetry in how things show themselves. And so where we're going with next on this is, I think Mike Gore and John McKay and a bunch of people have pangenomes of a bunch of the canola types. We're making pangenomes of rutabaga and Siberian kale. Hopefully someday we'll bring all these together and have a big party. And then of course we're CRISPRing out, everybody else is doing oil seeds and glucosinolates. We're actually going after some other targets that nobody else cares about. So the future. So here's my future. This is my, Ed, this is your slide number 2 for you and friends. So maize subgenome assignments are problematic if they're just sorghum and maize comparisons. You need other genomes to fill in who the parents are to do it right. It's still biased fractionation, but it's going to be done differently. So in general, as a good systematist and phylogeneticist, move away from pairwise comparisons to phylogenomic comparisons. That's message number one, no matter what your omics. And then think about the modeling. So how would you model what deleterious alleles you want to go after using the framework of both phylogenomics and population genetics? And so we spelled this out in a 2018 paper in ARES. And then we've had a couple of some other thought pieces about this, both with Gaurav and AJB. Sarah Turner Hassong and Jeff Ross Bar and I talked about these evolutionary insights impacting plant breeding. So what, breeding 5.0? I don't know what we would call that. And then, and this is an older paper, but I actually reread it on the plane out here. Jacob Washburn and some of us wrote this paper on convergent evolution. And I think that there's some nice ideas in there about thinking about constraints. So I'll just end by acknowledging my funding sources, including USDA, which is new for me because I have friends here. National Geographic is new, so now I get to have music or something. I don't know what that means exactly for National Geographic. And I didn't talk about it today, but we have a small DOE grant, not DOD, on using Brassica as a low-dose radiation sensor, which actually we can just plug in with all the other stuff that we got going on. And JGI has given us a ton of data, and I didn't talk about an orchid mycorrhizal fungi project today. So these are the people that do the work. So I talked mostly about the work done by McKinsey, Holman, and these are new students who are working on various projects that have to do that follow up, mostly CRISPR and functional genomics. And I have had this great visiting scientist, Wan Zhijiang from China this year, who's been helping us with double haploids and CRISPR also. And what I'm most proud of our lab is our undergrads, because they come in as whatever, pre-meds or not knowing anything really, and they end up going to places in grad school all over the world doing cool stuff. So thank you. Last fun fact, if you didn't know it, pumpkins were really, really cool for people because like they used to carve turnips and rutabaga, and it was really hard and you got your hands all bloody. So our lab has an annual turnip carving party, rutabaga party. So this is last year, we'll do something like this year. But home much prefers to carve pumpkins to these. So, all right, so that's it. Thank you. Questions? I can handle it. Awesome. All clear. Clear as mud. All right, go ahead, Gara.
So you showed the two genes. One of them was, I guess, on LF, the least fragmented chromosome fractionated copy, and the other one had very low expression level and expression level conditions. So we know that in MF1 and MF2, there are many of the genes are fractionated. Is that so? The model that was presented was that Brassica is sending out all emergency responders when there is stress, but is it that it's just that gene is on the way to pseudogenization and it just retains some of the activities that were there before by chance, and so it's not a selected response by Brassica, instead it just...
So yeah, so this is, if Gavin Conant would hear, he would love your question. So we actually wrote a paper about noise, which is why you need the phylogenies. You have to distinguish noise, which I would say things on the way to pseudogenization versus things that are fixed by subfunctionalization, right? If you had true original four-cell subfunctionalization, you're stuck with them, right? You can't get rid of them. But if it's these things that it's kind of noisy and they're on their way out again, like you're still playing. So this is why I like your work on enzymes, because, okay, so in polyploidy, what does everyone want to do? They want to see polyploidy on the tree, and they want to map that where that polyploidy even occurs, you have diversification. No, you have polyploidy. Well, then evolution's got to tinker around and figure out how the enzymes are going to find their substrates, or figure out how they're going to do their homeostasis, blah, blah, and then you get a novel trait, and then they can diversify, right? So what's the time lag for that? So for me, thinking about AI or whatever, what's the time frame that I want to look at after polyploidy? Like, so how far is it? Like, is it what pathway am I looking at? Do I want to look at the mesopolyploid event, the alpha beta event, or the Brassica napus event? So I don't know what your enzyme you're working on or whatever, but for me, that's like where I would start with answering a question, or whether it's cardiac glycosides. Or, but most of these I think are fixed if they're like have really deep partitioning between tissues. And so the other thing we've been trying to model is, so Jen Weisskaber and I have been thinking about, well, if I really want to know about the glucosinolate module, how do I spread the dollars between different species, accessions within a species, and tissues and time points, right? And so like, okay, well, let's think about that. If you have some prior idea about how big the module is, you can figure that out, right? Or what the gold standard stress is that's going to do 5 things instead of 1 thing. Copper sulfate instead of methyl jasmonate, maybe, or I don't know, I'm making it up. But anyway, so some of these are noisy, right? But that's when we look at a pop. So I showed you a canonical example, but in a PopGen framework and maybe some other things, like they don't look as clean as that. Oh, maybe the pseudogenizing in that Arabidopsis cole line, but not in Arabidopsis layer, or I don't know if that answers your question, but we're just looking at a snapshot, right? Which is why you need to test nodes. So, Chelsea.
So I'm still thinking through the process of the de-domestication, and I really loved your example and the way you flew through like 1,000 ideas all in like a couple of slides. That's fantastic. But thinking about that, so one thing you notice is that you get these trends. You get a trend towards woodiness. You get a trend from the end. So you can start thinking about the things that cause the domestication by what is the release of that.
Exactly.
Environment.
Constraint again.
And so do you think, and the woodiness was on islands where you see plants growing woody anyway. So is there any, are there any insights that are jumping out from that that you hadn't maybe thought of that are providing an idea of what that canalization process is and why you don't see such overwhelming diversity of forms given the overwhelming genetic potential and variation that's out there?
Can you repeat a short version of a question for people on Zoom?
Sure.
Sorry.
Is it, do we see canalization?
Sure. Yeah, do you see any, are there any sort of phenotypic insights coming out of the de-domestication
Yeah.
that provide ideas about variation in genetic?
Yeah. So the question is, do we see any patterns of de-domestication that may tell us about some central process?
Yes.
Okay. We don't know yet. We're just starting. So for me, though, again, it's like, what's the scale of the problem? Are we trying to figure out, okay, in Brassica, how does Brassica become woody? Or do we want to look at like, well, how do all flowering plants become woody? And look at Asteraceae also and some other ones, right? And so there's a project in Europe where people are looking at woodiness evolving both naturally on islands and also these kind of de-domesticated types. So I'm curious, things that share, so just to narrow the question a little bit. So things that share the same hexaploidy in the Brassicae, what's the difference between things that were perennial that then became more woody versus things that were domesticated into herbaceous and now become woody? What can I learn about woodiness from the difference between those two, right? And are there constraints? Is it always the same little switch? Is it the same little transcription factors over and over again? So that's the question. I don't know the answer. Any other questions I can't answer? Or I can look philosophical like I know the Jeff. Oh, one more question, then we got to go.
Okay, sure. So, you know, you framed the Brassica versus Arabidopsis thing, yeah, with the helicopter and the boat and all that kind of stuff. It's nice.
Is that okay?
Yeah, I like it a lot. But Arabidopsis had that opportunity too, or an earlier Brassicaceae,
yeah,
right? And clearly it didn't go that same route.
Right.
So what's your thought about the sort of the negative aspects of maintaining some of those options, maintaining extra copies of genes?
Well, there's clear, I mean, you mostly have gene loss due to drift, and clearly, I mean, we all know that genome size is a huge cost, right? I mean, Arabidopsis' entire life cycle occurs in the time span that Lilium does meiosis. So, you know, if I had to choose, fast and weedy, yeah, but I just think trying to broaden your question or thinking about constraint. I mean, there's different ways to have variation too, right? So like splice variants or, you know, transposons, blah, blah, blah. So I'm just focusing on my own research program is just trying to focus on tandem versus polyploid duplicates, but there's a lot of ways that variation could happen. And certainly there's genomic. So when I think of genomic architecture, that's what I'm thinking of is this kind of network architecture. And so what did Arabidopsis give up by giving up those other networks, right? I don't know. Doesn't have a helicopter, but it has a pretty fast speed. You know, it has a pretty fast boat, right? So, can I poorly answer another question? I've done two. I'm 2 for 2 on 0. Or is it time to go? Time to go. Sorry. Time out. I'll be here. Okay.
This has been a production of Cornell University, on the web at cornell.edu.